In this video, we will be discussing the mechanism of heart excitation and contraction. The heart beats around 100,000 times per day, pumping blood throughout the body. But have you ever wondered what makes it tick? The mechanism of heart excitation and contraction is the remarkable process that turns electrical signals into life-giving heartbeats. It's a tiny but vital process that happens in every heartbeat. Let's dive in and explore how it works. The mechanism of heart excitation and contraction is a complex process that involves the coordinated effort of multiple cellular and electrical components. First, we will discuss the events of heart excitation and then we will learn the heart contraction. The heart excitation, also known as cardiac excitation, refers to the electrical impulse that triggers the heart muscle to contract. It is the spark that makes the heart pump. The heart generates a spark, which is initiated by the SinoAtrial SA node, then travels through the Atrioventricular AV node, followed by the bundle of His, and finally reaches the Purkinje Fibers, causing the heart muscle to contract and pump blood. The word SinoAtrial is a combination of two terms. Sino refers to the term sinus, which means inlet. It also refers to sinus venerum, a small cavity in the right atrium of the heart where the SA node is located. The term atrial refers to the atria, the upper chambers of the heart. So, the term sinoatrial literally means pertaining to the sinus and atrium. In the context of the heart, it refers to the SA node's location in the sinus venerum of the right atrium. The SA node, also known as the sinus node or pacemaker, is a small group of specialized cells located in the right atrium of the heart. It plays a crucial role in regulating the heart's rhythm and contractions. It generates an electrical impulse, action potential, approximately 60 to 100 times per minute. This generation of electrical impulses occur through a coordinated process. It starts with the opening of sodium channels, allowing sodium ions to rush in and depolarize the cell. This rapid depolarization triggers an action potential, fueled by a slow influx of calcium ions. As the action potential peaks, the sodium channels close, and potassium channels open, allowing potassium ions to leave the cell and repolarize the cell. A slow depolarization, known as the pacemaker potential, then occurs, gradually building up to the threshold potential, which triggers the next action potential. This cycle repeats, generating electrical impulses at a rate of 60 to 100 times per minute, setting the heart's rhythm. AV node. The atrioventricular node is a small specialized group of cells located between the atria and ventricles in the heart. It plays a crucial role in regulating the heartbeat by coordinating the electrical signals between the atria and ventricles. The AV node is often referred to as the relay station or gatekeeper of the heart, ensuring that the electrical signals are properly transmitted and coordinated to maintain a consistent and efficient heartbeat. The impulse travels to the atrioventricular node, which delays the signal for about 100 milliseconds. It relays the impulses to the ventricles and trigger their contraction. Bundle of His. The bundle of His is a group of specialized fibers that transmit electrical impulses from the AV node to the ventricles, coordinating their contraction. It is located in the atrioventricular septum and divides into left and right branches, which then split into smaller fascicles, reaching the ventricular muscle cells. This critical highway ensures efficient and synchronized contraction of the ventricles, regulating the heart rate and rhythm. It plays a critical role in the heart's electrical conduction system. It's named after the Swiss cardiologist Wilhelm His Jr., who first described it in 1893. Purkinje Fibers. Purkinje fibers are specialized conducting fibers that emerge from the bundle of His, forming a network of strands that spread throughout the ventricles. These fibers transmit electrical impulses to the ventricular muscle cells, triggering their contraction. Purkinje fibers enable rapid conduction of electrical signals, ensuring coordinated and efficient contraction of the ventricles. They play a crucial role in regulating the heart's rhythm and pumping action, making them a vital component of the cardiac conduction system. Purkinje fibers are named after the Czech anatomist and physiologist Jan Evangelista Purkinje. We summarize the heart excitation events as the heartbeat starts with an electrical impulse from the SA node, flowing through the AV node, bundle of his and Purkinje fibers, causing cardiac muscle contraction and pumping blood. Let's dive into the topic of heart contraction. This complex process involves several phases. The depolarization is the initial electrical impulse generation phase. After that, the calcium is released, which triggers the muscle contraction. Following the calcium release, the heart muscles tighten, which then cause the ventricular contraction, which then pump blood out to the body. After the ventricular contraction phase, the ventricles get relaxed and become refilled. Depolarization is the term that can be broken down into "De" means removal, and polarization refers to the state of having a distinct positive and negative charge. So, depolarization specifically refers to the rapid change in the electrical potential across the cell membrane, from a negative charge to a positive charge, which triggers muscle contraction. It is a critical step in the heart's excitation-contraction cycle and occurs when the electrical impulse from the SA node reaches the ventricular muscle cells, causing a rapid change in the cell membrane potential. This change allows positively charged sodium ions to rush into the cell, while positively charged potassium ions leave the cell. As a result, the cell's electrical charge shifts from negative to positive, triggering the release of calcium ions from the sarcoplasmic reticulum. This flood of calcium ions then triggers muscle contraction, allowing the heart to pump blood out of the ventricles. In essence, depolarization is the spark that sets off the heartbeat, transforming electrical energy into mechanical force that sustains life. In short, the electrical impulse reaches the ventricular muscle cells, causing depolarization. After depolarization, calcium ions are released from the sarcoplasmic reticulum, a type of smooth endoplasmic reticulum found in muscle cells. This calcium release is triggered by the rapid depolarization of the cell membrane, which opens voltage-gated calcium channels. As calcium floods into the cell, it binds to troponin and tropomyosin, causing a conformational change that exposes the actin-myosin binding site. This allows myosin heads to bind to actin filaments, initiating the muscle contraction process. The sudden increase in intracellular calcium concentration is the key event that links electrical depolarization to mechanical contraction in the heart. The calcium release triggers a synchronized contraction of ventricular muscle cells. Calcium binds to troponin, exposing the actin-myosin binding site, and myosin heads bind to actin, generating force through a power stroke. As multiple sarcomeres contract, the ventricular chamber decreases in volume, and pressure increases, propelling blood out of the heart. The coordinated contraction of ventricular muscle cells, fueled by calcium release, ensures efficient pumping of blood, maintaining cardiac output and perfusion of vital organs. This precise regulation of calcium-dependent contraction enables the heart to adapt to changing physiological demands, such as increased blood pressure or physical activity. After contraction, the heart muscle relaxes as calcium is rapidly sequestered away from the contractile apparatus. The sarcoplasmic reticulum and sarcolemmal calcium pumps remove calcium from the cytosol, reducing the intracellular calcium concentration. This decrease in calcium allows troponin and tropomyosin to return to their resting positions, inhibiting actin-myosin interaction and terminating contraction. As the ventricular muscle cells relax, the ventricular chamber increases in volume, and pressure decreases, allowing the heart to refill with blood. This precise regulation of calcium-dependent relaxation enables the heart to efficiently alternate between contraction and relaxation, maintaining a normal heartbeat and ensuring adequate blood flow to the body. The muscle cells repolarize, and the ventricles relax, allowing the heart to refill with blood. This process repeats with each heartbeat, generating a continuous cycle of excitation and contraction that pumps blood throughout the body. Now, we will be discussing how heart's electrical activity can be measured. An electrocardiogram (ECG) measures the heart's electrical activity, tracking heartbeat rhythms and aiding diagnosis of cardiac conditions. Atrial depolarization is the spread of electrical impulses through the atria, causing them to contract. It's the first phase of the cardiac cycle, triggered by the SinoAtrial node. The impulses travel through the atrial muscle, causing depolarization and contraction. This leads to the P wave on an electrocardiogram, ECG. Atrial depolarization prepares the heart to pump blood into the ventricles, ensuring efficient circulation. Ventricular depolarization is the spread of electrical impulses through the ventricles, causing them to contract. It's the second phase of the cardiac cycle, following atrial depolarization. The impulses travel through the ventricular muscle, causing depolarization and contraction. This leads to the QRS complex on an electrocardiogram, ECG. Ventricular depolarization is the main event in the cardiac cycle, pumping blood into the circulatory system. Ventricular repolarization is the recovery phase of the ventricles, where they return to their resting state after contraction. It's the third phase of the cardiac cycle, following ventricular depolarization. During repolarization, the ventricular muscle cells recharge and prepare for the next contraction. This phase is represented by the T wave on an electrocardiogram. Ventricular repolarization is crucial for maintaining a healthy heart rhythm, as it allows the ventricles to recover and replenish energy stores, ensuring they're ready for the next heartbeat. So, an electrocardiogram, ECG, is a non-invasive test that records the electrical activity of the heart. It measures the electrical impulses that control heartbeats and displaying them as waves on a graph. This test is quick, painless, and provides valuable insights into heart function, helping doctors diagnose and monitor heart health.